So today's video is going to focus on viral reproduction and viral infections. So it's kind of two videos combined into one. So we'll start first though with how the virus actually reproduces, because that is also essentially how it's infecting us. Okay, so a few reminders on viral structure. Remember, all virus, regardless of kind, right? We have lots of different kinds of examples of viruses here. Helical shaped ones, polyhedral, spherical, these more complex ones like the bacteriophage here. They're all going to have some kind of DNA or RNA in the middle, though. You can see that in each of these. And they're all going to be surrounded by that capsid or protein coat. And then some of them will have various surface markers. You can see on a few of these images here that are going to help it attach to particular receptors and to particular cells. So let's take a closer look at once it gets inside the cell. Because like we mentioned before, it's got to make proteins to be able to make this capsid. It's got to make more DNA and RNA to be able to replicate its genetic material. And that's where your cell is going to come into play. It's going to essentially hijack your cell. So a virus essentially can have two life cycles, for lack of a better term, that it's going through. So let's look at this first one here called the lytic cycle. We've talked about this word before, right? Remember lysis, to burst, to explode, right? So in this lytic cycle, the virus is going to be bursting out of the host cell. Okay, so let's look at this example that we have here. So we have a host cell, which is this bacterial cell here, right? We can tell it's a bacterial cell because it's a prokaryote because there's no organelles in it. That DNA isn't in a nucleus. So the virus first needs to attach, and whether it uses those surface markers to attach or not, okay? And then the virus is going to enter its own DNA into the host cell. And it's going to break down the host DNA. See how it's breaking apart the host DNA here? So it's going to release its DNA into the host cell, and it's going to break down the host cell DNA, meaning now that the host cell DNA can't be used to make proteins. It's the intact viral DNA that the cell is now going to use to make proteins. So because the host cell DNA is broken down, step 3 can happen. Remember, when we synthesize something, we're making something. So this host cell is now doing all this work. This host cell is going to be who's doing the synthesis of the viral genomes and proteins, because that host cell is going to contain ribosomes to help make those proteins. That host cell will also contain various nucleotides, right? The pieces, remember, nucleotides are the monomers of nucleic acids. So it will contain the nucleotides that can be put together to make new viral DNA. So now this virus, the whole virus didn't enter the cell, but the DNA for the virus for sure has entered the cell. The cell has started making the parts of the virus. You can see how in step 3 here, it's all in parts, right? You can see the genetic material. You can see the capsid, the tail parts, right? So it's making it all in parts. And then it will use, again, host cell machinery to assemble the virus into complete viruses. Once that host cell is full of viruses, this is where the lysis part comes into play. These viruses are bursting out of this host cell. Well, based on what you know, right, about cells, if I punch through the cell membrane and I leave a hole in the cell membrane of this bacterial cell, this bacterial cell, this original host cell here, is going to die. This host cell has died now. It can no longer maintain homeostasis. It can no longer maintain its internal environment because its cell membrane has been so damaged by these viruses exiting. So the original host cell will die. So by the time it circles back around from step 5 to step 1 again, this is a new host cell and a new virus. These viruses that are released here will all go infect new cells. So as this occurs as this virus is spreading to more and more host cells, more and more host cells are being damaged. This is when we start to see signs and symptoms of illness. This is when we get a runny nose, a stuffy nose, a sore throat, vomiting. That's when all of that will start to happen, and they will, and that infection will start to show for us. Occasionally, a virus may not be going through a lytic cycle. It may be going through a lysogenic cycle. Okay, so a lysogenic cycle is going to be a little bit different. Maybe for some reason the environment isn't conducive to the virus at the time. So the virus will essentially hide. So in the lysogenic cycle, a big difference here is that it does not destroy the host cell like the lytic cycle did. So in this instance, the phage, the bacteria or the virus, again, we're using a bacterium as an example, the bacteriophage releases its DNA into the cell again. This time, the virus DNA, the phage DNA, joins with or integrates into the bacterial DNA. So the host cell DNA is not broken down like it was in the lytic cycle. The host cell DNA remains intact, and the virus DNA is incorporated in as part of it. So now, every time that host cell replicates, so that's what happened here. That's why we have two of them. The host cell has replicated, and that viral DNA is still in that host cell. Those cells may or may not have different properties than the original cell did because of the viral DNA in them, but it keeps that viral DNA intact. And so when the environment is now conducive to the virus, it will switch over to a lytic cycle. Okay? And then it can start causing infection and breaking those cells apart. So it will switch over to that lytic cycle. An example of this in humans would be the herpes virus or the chickenpox virus, which is a form of the herpes virus. But let's use cold sores as an example, right? That's a form of herpes. And cold sores come and go, right? Once you first get a cold sore, they don't go away, and you might not have one for a while, but eventually they'll come back again. Okay? That's because that virus goes back and forth between a lytic and a lysogenic cycle. When you have a cold sore, it's in that lytic cycle. You're super stressed. It's so your immune system's a little bit weaker, so the virus can manifest in that lytic form. And then when your body gets it back under control again, that virus just retreated back into this lysogenic cycle. So it can bounce back and forth between the two cycles. And we'll look at that on our next diagram. So in this diagram here, you can see how this is happening. This virus may go through this lysogenic cycle lots and lots of times before it ever switches over to the lytic cycle. And that's going to be determined by those environmental factors. So you can see it can bounce back and forth between the two, actively infecting you in that lytic cycle. So with that lytic cycle, we see symptoms very quickly within a few days to hours of infection. That lysogenic cycle, you may not see symptoms for months, weeks, years until it enters that lytic cycle again. Okay, so let's switch gears then to some particular viral infections. You don't need to memorize all these. This is just kind of an idea to show you that there are a wide variety of viruses that can infect us as humans, causing a wide variety of symptoms. Okay, so first a little vocabulary, talking about a pathogen. A pathogen is anything that can infect living cells. So virus, bacteria, fungus, they're all examples of pathogens. Antibodies are what we produce them. So we produce to help fight those. We talked about antibodies a little bit when we did blood types, right? And we were producing things that were the attackers, right? So antibodies are our attackers. Antibodies are, as we mentioned also with blood type, they're antigen specific, meaning like all protein-based things, because antibodies are protein-based, like all protein-based things, they have a specific shape. And so only certain antigens can fit with certain antibodies. As you can see, there's multiple antigens on the top here, but only this particular antigen can be attacked by that particular antibody. So the surface markers on the virus will make antibodies. These are antibodies, the darker ones here, will make antibodies in response to those surface markers to be able to find and attack the virus. So some examples of viral diseases, we have coronavirus, as I'm sure you guys are now well aware of. Rhinoviruses, those are usually examples of the common cold virus. Herpes virus that we've mentioned a little bit earlier when we were talking about reproduction. Hepatitis is viral. Measles and mumps are viral. Rotavirus, if you have young siblings that are in a daycare, they might talk about rotavirus. Rabies virus with animals and pets. HIV. Some of you may have heard of HPV, which is one that you may or may not get a vaccine for as you reach about ninth grade. Smallpox virus, you've probably never heard of because smallpox has been eradicated. So just again, showing we have this wide variety of things that can be caused by viruses. So how do we get better if we get a viral disease? Or how do we not get the viral disease, hopefully, in the first place? So one thing I want to be sure we're well aware of is that antibiotics, they don't work on viruses. Antibiotics, right? Bio is life. I'm against life. Antibiotics are working against the bacterial cell. They're particularly aimed at destroying the cell membrane or cellular functions that a virus doesn't do, because a virus doesn't fit that living thing criteria. It doesn't have a cell membrane that it can poke holes in. It can't disrupt the making of ATP. So antibiotics don't work for a virus. That's why you don't get antibiotics for a cold or for the flu. You don't get antibiotics for coronavirus. You could possibly get antiviral drugs. Antiviral drugs, as a general rule, they work in one of two ways. They try to prevent the virus from attaching to the cell at all, to the host cell, or they try to disrupt the viral life cycle. The struggle with antiviral drugs is that all these viruses are so very different that it can become very difficult to develop drugs for each of the different kinds of viruses. Viruses also mutate very quickly. And so if the virus is constantly changing, the drug has to be able to change with the virus, which also makes antiviral drugs more difficult to develop. Vaccines, as a general rule, vaccines you're inoculated or given, right? That's given the shot. So it's introduced into the body, usually a dead or a weakened form of the pathogen. So you receive this shot that contains a little bit of dead or weakened form of the pathogen. The graphic here also talks about mRNA, which is, I know, a current hot topic with coronavirus and it being an mRNA vaccine. All of these things are doing the same basic job. They're essentially teaching your body how to make antibodies for the disease, because your body has to learn to make antibodies. That's why once you get, for instance, chickenpox, you shouldn't get chickenpox again, because your body learned how to make the antibodies for chickenpox. So if you're exposed to that virus again, it'll make them super fast. Second exposures, our body works much faster than primary exposures, because on the first time we have to learn what to do. The second time we already know, and we just need to crank it out. So the vaccine is going to teach the body to recognize the virus. So whether that's the dead or the weakened form, or if it's the mRNA form, the mRNA will enter your cell. It doesn't go to your DNA, because we know that's not the direction things flow. They flow DNA to mRNA to the protein. And then so it'll use your own cells to make the proteins that the virus contains. So not the DNA, just the proteins that the virus contains. and then therefore your body can recognize the virus. So you didn't have to get a dead or a weakened form of the virus. You got this mRNA, your body made proteins, your body then can make antibodies, learn how to fight those particular proteins. And then if you're exposed to the real full-blown virus, your body can quickly respond to it. So when we have what's called an outbreak is going to occur when there was no immunity essentially to begin with, and the disease will spread very, very quickly. Okay, we've seen this happen with various diseases. An outbreak happened with coronavirus. There was some large flu outbreaks in the early 1900s. These are the outbreaks, are what they're talking about when we talk about how when settlers came to the New World, the Native Americans had never seen some of the diseases they brought with them. And so those diseases, the Native Americans didn't have any immunity to fight those diseases. And so those diseases would wipe out large populations within the Native American communities. An epidemic versus a pandemic. So making sure we get our vocabulary down real quick because they're a little bit different. An epidemic is going to be an outbreak, but it is in a large area or a region, where a pandemic, pan means all, a pandemic is all over the entire world. That's why coronavirus was a pandemic, because it was all over the entire world. It was not localized to just the United States or just Europe or just a certain region of Asia. Okay, so we're going to look at one particular example of a viral disease of human immunodeficiency virus, or HIV, because HIV is very specific and it's a little bit different. HIV is the virus that causes AIDS, so it can lead to acquired immune deficiency syndrome. It does that by attacking these helper T cells. You'll see in this diagram here, the helper T cells help eventually make antibodies. They help eventually make macrophages, which are cells that are large cells that will essentially eat other cells. So they can eat infected cells. They can eat virus or bacterial particles themselves. So they're going to help you fight infection. And then killer T cells, which will kill infected cells, as their name implies. So helper T cells, you'll see, are playing a role in doing these various things. And so somebody that these helper T cells are infected and they're not working properly, they're not making appropriate antibodies, they're not making macrophages, so they're extremely open or susceptible to secondary infections like pneumonia, which somebody with a normal functioning immune system would be able to recover from. Somebody with AIDS may not be able to recover from that. So you can see on the right here how this HIV works. So HIV is what we call a retrovirus, retro being backwards or behind or in the past. So HIV actually has RNA in it, which RNA can't make, the RNA is actually going to be worked backwards into DNA. So now that DNA can go into the host cell genome. The RNA can't be incorporated right into the host cell genome because the host cell uses DNA. So the host cell genome is going to incorporate this new DNA that it made. Now the virus hijacks the cell and does transcription and translation to make more viruses. So that's a little bit different in the sense that HIV doesn't have DNA, it has RNA, but then can actually use an enzyme that is found, that will do reverse transcription and turn that RNA into DNA. Okay, so like always, questions written down, flagged in your notes so that we can clear anything up, so that we can get doing the hands-on application analysis, like doing the science.